in different science streams to present the advancement in their field of expertise. This seminar series is a collaborative effort between CUFSA and the Columbia University Faculty of Science. Welcome to the Distinguished Speaker Series of CUFSA.
Okay, good morning, good afternoon, and good evening from wherever you are joining with us today. My name is Vasundhara Fernando, representing CUFSA, and I will be the moderator for today's talk. First of all, on behalf of CUFSA Distinguished Speaker Series Subcommittee, I warmly welcome you all for today's talk. I would like to remind you that this talk will be broadcasted on our Facebook page and YouTube channel simultaneously. There will be a Q&A session, and I invite you to type your question in the Zoom chat or on our Facebook page or on the YouTube comment section at any time, and then we will direct the questions to our speaker at the end of the talk. All right. So today we have Professor Gomika Udugamasooriya from Department of Pharmacological and Pharmaceutical Sciences, University of Houston, and he's going to tell us about novel technologies that expedite the drug lead discovery process. Dr. Gomika Udugamasooriya obtained his BSc in Chemistry Special in 1999 from University of Colombo, Sri Lanka. He completed his PhD in bioorganic chemistry and biochemistry in 2006 at Wayne State University, Michigan. Then he completed a postdoctoral fellowship at University of Texas, Southwestern Medical Center, Texas, focusing on cancer chemical biology. So in his independent academic career, Dr. Udugamasooriya first joined as a tenure track assistant professor at the University of Texas in 2009. Then he moved to University of Houston, MD Anderson Cancer Center and Bayer College of Medicine in Houston, which is the world's largest medical center, and currently serves as a tenured professor of medicinal chemistry. The development of a unique high-throughput cell screen to identify highly selective drug leads for cell surface drug targets was one of the major milestones of Dr. Udugamasooriya's career. Professor Udugamasooriya is directing a multifaceted research program mainly focused on cancer drug lead discovery and diagnostic tool developments, funded by research grants from National Institute of Health, Department of Defense, Cancer Prevention and Research Institute of Texas, Innovative Research Award from Rheumatology Research Foundation, Owens Foundation, and various other sources. He has published over 40 research articles so far in world topmost journals, including Nature Chemical Biology, Journal of American Chemical Society, Clinical Cancer Research, and European Journal of Medicinal Chemistry. He also presented over 75 abstracts and gave about 50 invited seminars in US and worldwide. His novel drug discovery technology has so far produced 10 national and international patents. So recognizing his impactful and innovative contributions to research and success in patents and commercialization, he has been inducted as a senior member at the United States National Academy of Inventors in 2023, where only 435 senior members exist worldwide so far in the academy. Dr. Udugamasooriya is an editorial board member and a reviewer of many international scientific journals. He is a member of grant review panels in US government, National Institute of Health, Auckland Medical Research Foundation in New Zealand, US private foundations such as Keck Foundation and multiple international organizations. He received numerous awards, including Chancellor's 50 in 5 Scholar multiple times at University of Houston, Faculty Excellence in Research, NIH SPO Cancer Excellence Career Development Award, and his undergraduate career, he graduated winning the Gold Medal Award for Chemistry at University of Colombo. Prof. Udugamasooriya was the chair of the Graduate Education Committee at University of Houston. He has been advocating for many young scientists and engaged in numerous career-building seminars and workshops, both in the US and Sri Lanka. He is actively engaged in conducting seminars and workshops worldwide on cancer prevention and research, highlighting the value of practicing holistic medical approaches. Those include developing good eating habits, building psychological strengths, positive thinking, in addition to conventional cancer treatments. It's a great honor for me to read this extremely impressive bio and introduce Professor Udugamasooriya to our audience today. So without further ado, Professor Udugamasooriya, I kindly invite you to deliver today's talk on novel technologies to expedite the drug lead discovery process. Professor Udugamasooriya, over to you.
Thank you so much, Vasundhara, for that kind introduction and also for the invitation. Good evening to you all in Sri Lanka, and good morning to folks joining in the US and other places. So let me first share my slides. I hope you can see my slides clearly, right?
Yes.
Yes,
All
I
right.
can see. Yeah.
Okay.
Yes, it's clear. It's actually clear.
Right. So, so I'm going to talk to you about some of the novel technologies that I have been developing and applying in the last decade or two, about 2 decades in my research career. So first of all, it all began here, University of Colombo, where I did my undergraduate work, and also the CUFs are also very close to me because I was also a founding member of the CUFSA a few years back. So I really want to thank both CUFSA and also the Chemical Society of University of Colombo for inviting me to give this talk. And also, I cannot forget my undergraduate mentor, Professor Dilip De Silva, who actually really nourished me not only in academically and also help in a lot of things in the personal life as well. So I really want to thank you all. And then let's get into the topic right away. These three words, bench to bedside, has been very important. Discussed a lot in the biomedical field within last few years, and there are a lot of reasons for that. So what this means is how we can translate this basic research which produce this mostly the drug leads, and then translate that into the clinic as a real drug. So it looks very simple, but it is not. There are a lot of effort, and also there are a lot of concerns as well. So I'm going to talk to you those in next few slides. So first of all, let's see the whole process of this modern drug discovery pathway. So academics like us identify the initial drug leads that can be targeted into various biomarkers in various diseases. And then they undergo cellular level and also animal level, and also finally 3 clinical trials to produce a drug. So in order to get one drug, we have to actually input about 10,000 compounds. So remember, all these 10,000 compounds are active on the target, but there's only one become a drug. And also, it costs $2 to $3 billion, and it takes about 10 to 15 years. So when I was presenting this, like few years back, this was, we were even called 1 to 2 billion. Now this number is increasing, and it further increase, and I'll talk about that in a minute. So if you look at this graph, it shows the drug output over the years versus R&D expenditure. And you can see this black line shows the R&D expenditure, which really growing. We spend a lot of money on R&D, but the drug output is, it doesn't follow that. So this is a really big question I have been asking for a long time. Why is this? There's something wrong. And then recently, the Forbes magazine posted this article, which says almost half of the big pharma companies actually hitting the negative R&D productivity, and they actually spend $6.16 billion per drug approval. So within last 20 years, which is 2001 to 2020, big pharma companies increase actually their R&D spending by 6% annually, launching about 250 on drugs. But their efficiency is $6.16 billion per new drug. Actually, Forbes doesn't make this news. It's actually based on this eye-opening review published last year in Drug Discovery Today. So this is a huge amount, and this is the question. And also, CAS published this article recently. What are the challenges in the drug discovery? And they very precisely identified two major reasons. So one is the drug candidates can be incorrectly identified as safe for humans, but approved for clinical trials, and they fail there. And the other one is the opposite. The drug candidates can be incorrectly identified as unsafe for humans, but disqualified for clinical trials. They were not taken forward. So, if we look at this modern drug discovery process and how it happened, so usually what happens is the biologists identify the disease specific biomarkers, and then chemists and biologists come and develop drugs to target that biomarker. Then only we look whether it has off-target effects. So we identify these compounds targeting whatever we want to target the biomarker, and then only we think about whether it has off-target effects or not. So we spend like many years of time, billions of dollars, and then we find like more than 99.99 compounds has side effects, off-target effects. So this is one of the reasons contribute here, because we don't consider the safety of the compounds we identify at the very beginning. There are so many other factors also. This is one of the main reasons. So I have been thinking this for a long time, and in my whole research career, I was focusing on can we bring this normal cell or the off-target effect to the initial step of the drug discovery. So we can actually eliminate a lot of non-specific compounds and identify the most specific or compounds that has least off-target effects. So that's kind of my main theme of my research program. So basically, I'm trying to address this issue, which is sometimes, most of the time, we identify compounds that are not safe, that has off-target effects. Okay. Yeah, actually, this came like few months back, but I have been working on this for a long time. Okay. So we are targeting cell surface biomarkers. Two main techniques we use are actually, the first one is the compound class, peptoids. I will explain what is that in a minute. And then I developed this on-beat two-color cell screening assay to identify not only the compound that targets the biomarker, but it doesn't bind to any other cell surface molecule. So that's the key feature of my screening method. So in drug discovery, there are two major ways of doing that. First one is the structure-based rational drug design, which is we look for the target structure, especially the active site, and then I develop a compound that can bind to that. It's like cutting the key to the keyhole. The second method is actually the combinatorial high throughput screening. You develop large libraries of compounds and then expose to the biomarker, and then the biomarker picks the best one. So it can be used for imaging and drug delivery as well. So we are actually working on this combinatorial high throughput screening approach. So these are the three major clinically approved drug classes so far: small organic molecules, peptides, and antibodies. So the compound class that we are working on, peptoids, is actually derived from peptides. So this is the basic difference between peptide and a peptoid. I think most of you all know what is peptide, which is a chain of amino acids, natural amino acids. So the amino acids has this side chain on this alpha carbon, but in peptoids, it has the same backbone, but this R group is moved to the amide nitrogen. So because of this, these peptoids are serum stable, non-immunogenic, highly cell permeable, highly diverse. Synthesis is very rapid and economical, easy to optimize, reported as great protein binding ligand. So there are all the great characteristics naturally in peptides. And I was so lucky to interact with the inventor or father of the peptide, Professor Ron Zuckerman. And also my postdoctoral advisor, Professor Thomas Kodadak, has been considered second father of this compound class because he's the one who actually popularized this compound class of biologically amenable compounds. Okay, let's go to some little bit of chemistry, but after that I will be hugely focused on biological applications. So the synthesis is very straightforward. We use solid phase synthesis. We use the small resin beads, which has active centers ready to react. In peptide synthesis, we add one amino acid at a time to build the peptide sequence. So in peptoid synthesis, in order to add one residue, which is equivalent to an amino acid, we go through these two steps. Bromoacetic acid brings the two carbon, and then this bromine is substituted by any amine. And you can see the R group, the side chain comes from an amine, so it has huge diversity. So I mentioned we develop large chemical libraries. How do we do that? So we use this split pole synthesis. Very briefly, let's say we have three building blocks, three amines called A, B, C, and we take three reaction vessels and we couple A here, B here, C here as the first residue. There are a lot of beads, and then we mix them and then randomly separate them back to three vessels, and then each one has all these three representatives, and then we couple A here, B here, C here as the second residue. Then we already have 9 combinations. So you can see if you have more amines, more building blocks, more steps, we can go very high. So this is actually a compound library of 250,000 compounds. And uniquely, each of these reactions can be completed in microwave assisted synthesis protocol, so which can be done in less than 1 minute. So we developed this library within few days time. So you can see amino acid like side chains also can be used, but this chemical diversity comes into this side chain really make a big difference because they are the side chains that go and detect bind to the various biomarkers. So these compounds can actually recognize areas that natural peptides cannot recognize. This is another library of 400,000 compounds. So at the end of this process, we end up with one bead, one compound format. That means each bead has a unique sequence with a lot of copies. Different beads have different compounds. The standard method when I was thinking about developing my technology was practiced for about 2 decades. What it does is, let's say we want to target this protein, identify a drug lead for that protein. So it's been engineered with a tag and then mixed with the library. And if it binds to a compound, that bead get light up. So we pick it up and then identify the compound. So it become a drug lead for that protein. This is good, but once again, we don't know the specificity, whether it binds to any other biomolecules in the body or cells. So these are the limitations. So in order to address these limitations, I developed this new technology where I left the receptor in its natural condition in the cell and exposed the whole cell to the library, but with a very important control. So basically, that is without the receptor. So basically what I did was took a cell line, divided into two groups, and overexpress the receptor we want to target. So in this case, we use VEGF receptors as the model system. So when the tumors are growing, it cannot grow beyond few millimeters without the blood supply. So the tumors release this VEGF hormone, and then it comes and binds to these VEGF receptors in the nearby blood vessels, and then trigger signaling, asking these blood vessels to develop new blood vessels towards the tumor. So this is called angiogenesis, and then the tumor grows very rapidly. So targeting this angiogenesis is one of the pathways to attack cancer. So we use these VEGF receptors as the model system. So basically, in one cell group, I have the VEGF receptor expressing, the other don't. And then I did a very simple thing. I stain these receptor positive cells in red, the parent cells in green, mix 1 to 1 and expose to the library. Now, if we see a red cell bound bead under this condition, it means this compound on this bead binds only to our receptor. If it binds to any other thousands of molecules on the cell surface, they are found in the green cells, and we will see both bound. So we can eliminate them at the very initial screening. So these are actual fluorescence microscopic images. So this is actually hit, which means only red cells bound. And look at this. So this is these beads carry a compound that may binds to the VEGF receptor, but they binds to the other off-targeted biomarkers in the cell surface as well. So previously we didn't have this green control, and then what we do is we pick these also and move forward in the drug discovery process, and then later we throw them out as non-specifics, or it has off-targeted effects. So in this study, we screened 400,000 compounds, and we found only 5 of these, but we got about 5% of these both bound beads. So that kind of tally the normal statistics in the drug discovery. So finding 5 hits out of 400,000 is a very huge stringency, and we later found all 5 binds to the same hotspot. So this is actually a summary of that project. We actually improved the compound, and I'm showing in the therapeutic side, we published a lot of work on this from this project. In the therapeutic side, we have validated this all the way to preclinical levels, and then we actually attached contrast agents and then deliver them to the tumors to image them for the diagnostic purposes in both in MRI and PET. So this is a very busy slide. I'm actually summarizing the 10 projects here that produces 10 patented compounds from this screening. So this shows the this column shows the disease applications, cell surface targets, our reports in the diagnostic or therapeutic area, and this is kind of showing the pipeline, the progress so far from initial screen to hit validation to clinical and to the clinical level. So I'm very happy that all these projects are reaching in the preclinical area. Green means completed, and orange means that's where we are working on. So we have compounds and the applications in cancer, rheumatoid arthritis, sepsis, COVID-19. There are many other areas. From this, actually, I will briefly summarize about 5 projects in different areas. So when I go through these projects, considering the time, I will go through the data very rapidly. So if you have any questions, you can ask me at the end of the talk. Most of these are published as well. So in the first project, we actually applied this technology to COVID-19 situation. The SARS-CoV-2 virus infected through the nose, and it actually binds to this human ACE2 receptor to get into the cell. So basically, this virus S protein binds to this human ACE2 receptor and then get into the body. Now, everybody is going behind this virus, which is including vaccines, which is a moving target. It actually changes with the mutation. So then the vaccines get outdated, it changes. So um, what we thought, can we put a block here which can binds to the our side, human side, host base targeting. So this is very simple approach. And also this all the SARS viruses use this same mechanism. It has been known for 20 years. Why there's no drug that develop targeting human side? Because this is a very simple idea. There's a big reason. This S2 receptor is actually not for virus to come and bind. It actually governs our blood regulation. So if we touch here anything, it actually messed up our blood pressure regulation and it become toxic for us. So the idea here is actually develop nasal spray type of application. So this is the crystal structure of the ACE2 receptor and the virus S protein. So we are trying to put this molecular mask. We name it as a molecular mask, but we really have to make sure it doesn't affect the enzyme active site. So as I mentioned, this ACE2 receptor is important for regulating our blood pressure. So we shouldn't target here. So when we look at this structure, it looks the virus very cunningly touch a different area to get into the body because it doesn't cause any toxicity. So when I thought about this project, I thought in most of the standard drug discovery tools can develop compounds binding to this active site. So how we can target here? So what I thought was, how about if we mimic in the evolution when the virus come closer to this receptor, when it binds in the first time, it recognizes whatever the easiest or the best place that can approach. So my hypothesis here was if we mimic this process, we may find the same place. And I have the perfect model because we have these beads displaying compounds that can come and bind to these receptors. So basically, that is what I did, but with this two-color approach, which means whatever the compound we find will guarantee not only bind into the virus binding site, most probably, and it doesn't bind to any other cell surface molecules, so it's highly selective as well. To cut a long story short, we found 2 compounds, and then we showed that it can uh, pick the receptors, and also it binds in the low mid nanomolar level. And also we showed that it actually blocks the virus S protein binding to the human ACE2. And then we also showed that it actually blocks the virus getting into the cells, including the mutated versions. And then we came to the very important thing, which is there are compounds actually reported that binds to the ACE2, but none has become a drug because it may affect this active site. So we really wanted to make sure this compound has no toxicity. So this is in multiple different cell lines, the cell cytotoxicity assay, no toxicity. And also we showed that it doesn't affect the ACE2 receptor protein. And also very importantly, we showed that this compound actually doesn't affect the enzymatic activity. So it is not toxic for us. And then we did some docking studies, and also we found that our compound actually bind into the same binding pocket, binding area where the virus touches. And now actually, it's actually the compound is in the animal studies, and we did those studies, but the pandemic went away. So, but I want to share this very interesting story. So this is actually Grammy Award nominee American artist Aloe Black. So when we published this first paper, he saw our novel approach, which is host-based approach, and then he contacted us and then started funding us. So he actually already funded like more than $500,000 in these projects. So we actually taking it into the next levels. So I just thought to share this story because it was a very interesting development. Okay, so as a summary, what we have done here was we actually develop a compound that binds to the human ACE2, and then it blocks the SARS-CoV-2 virus bind and getting into the body. So it's actually we call molecular mask. Okay, so let me jump in to talk about some of the projects in oncology, in cancer area. That is actually my main area. So in cancer, there are a lot of complexity and a lot of troubles here. So when the targeted drug discovery was initiated, everybody thought we can actually finish cancer. But if you think about targeting, here actually I'm showing the heat map of various receptor expressions. So they are very heterogeneous. In cell to cell, these targets are different. So even the best chemists come and develop a drug to target this receptor. It is not present in every cell. So that's one challenge. And then the signaling cascades are very complex. If you block in one place, it actually still the signal can bypass. So that's another challenge. And also there are like very stubborn a cell population called cancer stem cells, and they can actually survive standard therapy and bring back the tumor. So that's another big challenge. And then there are no perfect way to detect cancer in real time because we use different molecule to detect, and that has different PK/PD biodistribution properties. So these are some of the areas that in my lab we are trying to address using this technology. So if we go back to this technology again, uh, in the conventional process, biologists identify the compounds, and then chemists come and develop uh, drugs for those biomarkers, and then we look for the normal effect on the normal cells. So in my first technology development, I brought this factor also here, and then try to identify compounds that only bind into this biomarker we want to target that doesn't bind to any other cell surface molecule. I want to say actually it doesn't solve the whole problem because there are many different cell types, but I'm talking about at least to a certain level, we can reduce this off-targeted effects. And then I thought about applying this in a very unique way. So if we think what it does is it simply look at the differences between two cell surfaces. So then I thought, how about applying this in a totally unbiased manner? So the idea is we go back from molecular level to cellular level and compare two cell surfaces, for example, disease cell, cancer cell versus normal cell. And there are differences. And then we identify these differences in an unbiased manner. We don't know what it is, but what it does is it identifies compounds targeting something specific for cancer. And it can be a protein, lipid, carbohydrate, anything. So that's the cool thing here. We can identify new targets, new biomarkers in the disease condition as well. Okay. So the second project I'm going to talk to you is actually targeting cancer stem cells. So as I mentioned, um, these are the stubborn cell type found in the tumor. So typically, the normal cells can only divide. Like for example, skin cell can produce only a skin cell. But these cancer stem cells not only divide, they can also differentiate. They can differentiate into different cell types. So that's why they are very problematic. So in tumors, some of these stem cells can become cancer stem cells. So then it actually produces various type of tumor cells. That's one of the reasons we have very heterogeneous mixture. In the standard therapies, most of the cancer cells can be eliminated, but these cancer stem cells are stubborn. They actually remain and they bring back the tumor. So the idea here is to target these cancer stem cells so we can go for total regression. But the problem is the biomarkers are not well understood in the cancer stem cells. There are actually, but not very well defined. The biology is very complex. So this is where I thought about applying my unconventional approaches, which is totally screen in an unbiased manner. So what we did here was using commercially available kits, we separate cancer stem cells from remaining cancer cells from the same tumor. So the genetic background has to be same patient, same tumor. We separate the cancer stem cells and the remaining cancer cells. And then stain cancer stem cells in red and pair the remaining cancer cells in green, mix 1-to-1, expose to the library. And if we see a bead, only red cell bound bead, that means the compound on that bead binds to something specific for cancer stem cells, which is not found in the other cancer cells. So we did that say, and we found compound, and then we showed that it binds to the cancer stem cells, and this cells we pick has the stem character. We showed all of those, and then we don't know what is the target. So the way we identify the target is we let the compound to bind and trigger photo cross-linking, develop a covalent bond between our compound and the protein and pull it out and identify using proteomics approaches. And we did that and we found the protein as the plectin. And we showed that I'm not going to describe all these details. We confirm everything. It binds to the plectin, but there's a huge problem. This plectin is actually found in every cell, but internally. And it's actually not a conventional drug target. It's actually a structural protein. It actually connects these filaments. So at this point, we thought we messed up because our target should be on the cell surface. And then we found this very interesting paper, which showed unexpected gain of function for the scaffolding protein plectin due to mislocalization in pancreatic cancer. So what happened is this plectin protein actually come to the cell surface. So we found that phenomena happen in cancer stem cells, so which is very interesting. So this is actually we report this as a new biomarker for lung cancer stem cells. And then we showed that it is indeed on the cell surface. Actually, if you knock down, it affects the stemness. This is standard colony formation assay. And also, if you knock down the plectin, it actually affects the cell migration, which is connected to the metastasis. And also, if you look at the patient data, this is patient survival. High expression of plectin actually poorly correlated with the patient survival. So that also really interesting. And then actually we look at the activity, we further improve the compound, and we showed that it very specifically kills the cancer stem cells. And interestingly, it doesn't touch the remaining cancer cells. If you can remember, in the initial screen, we were really targeting cancer stem cells. So it doesn't target the remaining cancer cells. So this is very high selectivity. And also we showed that it actually destroy the colony formation, which is a standard assay used to validate these stemness. And also we showed that our compound actually blocks the cell migration, uh, again, part of the metastasis. And also in vivo studies, we showed that our compound can actually reduce the tumor size without any toxicity. No body weight has been changed. So there are a lot of work done on this study, but I'm just summarizing. So we did the two-color assay to unbiasedly target cancer stem cells, and we found new biomarker for the cancer stem cells. And then in the third project, we were trying to do again the complete unbiased approach. We did here a very simple thing. We took cancer cells and the normal cells from the same patient, same lung. So the idea here is this is a cancer cell. There should be some differences on the cell surface compared to normal cell, and that is what we are going to unbiasedly pick. So we stain the cancer cell in red, a normal cell in green, mix 1 to 1 and expose to the library. So we are looking for only red cell bound bead. So that means the compound on that bead binds to something specifically found in cancer cells. So we did the assay, we found the compound, and it actually pick only the cancer cells, not the normal cells. And then we don't know the target. So we were trying to cross-link and pull down the target. Uh, it didn't work. That's good news and bad news. So the bad news is we may don't know what is the target. But the good news is it may not a protein. So actually, to cut a long story short, what we found was this compound binds to a lipid called phosphatidylserine. So usually this lipid found in the inner layer of the normal cell. And then in cancerous situation, it comes to the surface. So this is the difference between cancer cell surface and a normal cell. Actually, this lipid come to the surface to tell the natural killer cells to don't kill me. It is a evading mechanism that tumors use to uh, protect themselves from the immune system. So this graph shows my collaborator found this compound, our compound binds very, very, very specifically to this phosphatidylserine lipid, and it doesn't bind to the normal lipid found in the cell surface. So it's a very safe and highly specific compound. And interestingly, this lipid flipping happen in almost every tumor. So I told you the protein biomarkers are heterogeneous. So we cannot develop a common drug that targets every cancer. But this lipid flipping is reported in every tumor. So this can be a global biomarker for drug discovery as well. And then in the activity side, we showed our compound actually very specifically killing cancer cells, but not the normal cells. And also in the uh, animal studies, this compound very, very specifically bind to the tumors, not the control. And then I'm here showing one of the tumor burden assays in animals. So this is the tumor volume versus treatment. The control tumor grows very rapidly, and our compound actually blocks the tumor growth comparative to this docetaxel, which is a chemotherapy. But the cool thing here is the combination with the chemotherapy. It doesn't even let the tumor to grow. It has been known that this chemotherapy actually increases the flipping of this lipid to the surface. So we have more and more target. So in this project, again, if you summarize, we compared the cancer cell versus normal cell and targeted unbiasedly, and we found a very unique lipid that comes to the cancer cell surface compared to normal cells. And then we have been validating this compound as another drug candidate. Okay, in the next project, we apply this in a very unique way for diagnosis focus. Now, this graph shows the tumor growth, which is at the beginning it is slow, but with the blood vessel formation, which is called angiogenic switch, the tumors grow very rapidly and go to the death of the patient. Unfortunately, the current detection threshold is after this angiogenic switch, so which is very close to the death of the patient. So can we bring this down? Can we identify tumors early as possible? Of course, everyone tries to do this. So that means we need to target these earliest biomarkers. So here I'm showing a normal cell moving through various steps to become a tumor. So early diagnosis means we need to identify targets that found in very early tumors. Again, the conventional approach is to look at, let's say we have 100 cancer biomarkers known. And then what we do is we try to find how many of them express in the early tumors. That's how the conventional approach is done, which is again biased. So what we thought was, how about if we take a primary cell line and convert it to the tumor and then identify these new biomarkers appearing as this transition happen. So this is Dr. John Mina at the UT Southwestern Medical Center. He's the director of the Cancer Center. So he actually developed this very unique cell series, which he took primary lung cancer cells and then step by step introduced these key mutations to the three key mutations that are important in cancer initiation and progression, and convert primary cells to the fully cancer cells. So my hypothesis here is when these mutations happen inside the cell, it starts changing the cell surface as well. So that means new biomarkers start appearing. So what does this mean? For me, these are cell pairs. We can compare normal cell to these initial levels, right? So that is what we did. And actually, to begin the project, we compared the normal cells to fully transformed cells. So the idea is there are new biomarkers appeared. We can pick them as they appearing. So we stain the normal cells in green, these transform cells in red, mix one to one and did the assay. And we found some of the compounds and showed that it only binds to the cancer cells. And also, again, we don't know the target. As I mentioned previously, we let the compound to bind, cross-link, and pull down and identify the target. So in this case, actually, we found vimentin protein, which is already known, but it is known in the latter part of the tumor progression, which is important in the metastasis, EMT process. But what we found was this is also expressed in the very early stages of the tumors, very, very early stages. And then once we have this early target and also the compound that can target, it can go to diagnostic applications and also therapeutic applications. So in the diagnostic applications, we attach contrast agents, the imaging agents, and deliver it to the tumor to light it up. So this is actually showing IHC studies compared to the commercially available antibody. The first four are tumor cells, and this is normal cell. First, actually, the antibody, and then we have our compound also doing the same kind of staining in this tumor slides. So it can be used for the early diagnosis. And we are trying to develop early diagnostic tests, which are very early stages. I'm not going to go into the detail. And also we found that this compound has therapeutic value as well. So we took cell lines, representative cell lines that actually we maintain high expression, moderate expression, and low expression. What we found was our compound is very effective in the killing these we maintain high expressing cells compared to the low expressing cells, and it doesn't affect the normal cells at all. So here I'm actually showing again the blocking the colony formation, cell migration. There are so many studies that I'm just trying to summarize the project. Once again, what we did here was we convert normal cells or push normal cell to become cancer, and then we try to pick the drug targets appearing while this process happens and then identify this vimentin as a biomarker for early diagnosis. Okay, so let me talk about the very last project, which is in the different area targeting sepsis. So actually, this is my one of my collaborators, Dr. Bingo. He brought this idea to me. So what it has been reported is when the bacteria or viral infection happens, this IL-15 overexpress, and when it binds to the IL-15 receptor, this triggers a very huge storm of immunogenicity. So as a huge immune response happen, increased T cells, NK cells, TNF alpha. So what basically happens here is when the infection happens, there's a huge immune response. And you all know when it go to the maximum levels, we call it sepsis condition. So when a patient come to a sepsis condition, doctors has only few hours to save the patient. And very unfortunately, there are no approved drugs to target sepsis. This is a very complex, very difficult area. And also even sepsis research is now considered graveyard because if you can remember the drug discovery process, there are a lot of compounds came in different ways. There are hundreds of clinical trials fail because this is very complex. And in this case, once again, we thought of not going behind the actual bacteria or virus. We actually thought of targeting, take care of our side. So we thought, how about if we try to block this IL-15 binding to the IL receptor? Because this is a receptor found in cell surface, so we can apply our assay and develop compounds that can bind here. That's what we did. We again compare IL-15 expressing cells to IL-15 lacking cells, stain the IL-15 expressing cells in red, the parent cells in green, mix 1-to-1, expose to the library. And then we look for only the red cell bound beads, which is IL-15 binding compound. And we found actually a compound, we optimized it. We did a lot of studies. I'm showing only 2 animal studies in the advanced level. We did. So the first study, actually, we first gave the compound to mice and then did the infection and monitor them. So in this first panel, we are video showing the animals without the compound. So actually we use Pseudomonas infection, which is very common in the clinic. So you can see the mice are extremely sick. Actually, they all died within hours. And this one is actually the same type of mice given with our compound. And you can see they are they survived and also they are very active. So this is a hugely interesting observation. And this is actually the murine sepsis score, which is very different with the control mice and then the treated mice. And then you may say, okay, this is compound was given early. So in clinic, what happened is sepsis happened first, and then we have to give a drug. Of course, we did the same assay in that condition. So we first infect the mice with Pseudomonas, and then in 4 hours and 48 hours, we did 2 treatments. And this video shows the 2 different conditions. Now, within 4 hours, the mice with the Pseudomonas infection, mice were very, very sick. And you can see it in the left panel. And in the right panel, actually, after giving the compound, in 24 hours, they all survive. They are living in a very active condition. Actually, in the survival curve, we lost 1 mice. So with the previous study, we did only 2 doses. If we gave the 3rd dose, we could have gone for 100% survival. So I have spoken about several different representative projects that were developed from this OBTC screening. So I'm going to summarize again. So what this assay does is it actually find in 2 levels. First, actually, it find compounds to our interested drug target, and the compound doesn't bind to any other cell surface molecules. Very high specificity, which is saving time and money on the drug discovery validating process. And secondly, I apply this in a very unbiased fashion, comparing disease cells versus normal cells, and then identify new biomarkers specifically found in those disease cells as well. So there were a lot of proteins we targeted, and also we were able to target lipids, and that's another unique thing, because lipid carbohydrate targeting is very difficult in the standard drug discovery process. There are not much tools are there. And I didn't talk at all about the imaging applications also we do. And also we try to combine those to develop compounds that has both therapeutic and imaging capacity built in. So that means we can monitor the drug activity in real time. So in cancer, this is very important application. Okay, finally, I want to thank everyone who supported my work. First of all the group members. So what I do here is sitting in this office and make sure we have grants and other things, but they are the real people who did the work on in the lab. And also I want to really thank all these collaborators from our university to many other universities all around the US. And finally, I want to thank all these funding agencies from NIH, Department of Defense, and many other agencies, and also our company, which I mentioned, Allo developed. So without funding, we cannot do anything. So I'm sorry, actually, I went really, really fast, but I want to thank CUPSA and also the Chemical Society at University of Colombo also for inviting me. And thank you all for listening to my talk.
Thank you, Prof. Udugamasooriya, for the great informative talk. And it is open for questions now. And until you get your questions ready, let's go through some of the questions posted onto the Zoom chat. So Angelo Gunasekara has 2 questions, and I'll read the first questions out to you. The first one is, well, there is a compliment. First, excellent talk, Gomika, and very innovative method. I have 2 questions. Why don't peptoids show immunogenicity when peptides and other proteins show this all the time? And if you want to take the question, I can give the second question later.
Okay, so that's an interesting, excellent question, because to be honest with you, we don't know what is the exact reason, but we have shown the peptoid compared to the similar peptides and proteins, it doesn't produce the immune signal. But I think one reason is, when the peptides proteins get into the body, it actually being processed and displayed for the to produce the immune as an epitope, produce the immune response. But the peptoids cannot be processed like that. probably that may be the reason. It cannot be chopped and presented in MHC cells, so probably that may be the reason. But we really need to explore and study why it doesn't. It actually flies under the immune radar.
Okay, thank you very much. And the second question is, there is a possibility that peptoids may not work all the time as a drug. Do you have approaches to use this method for small molecular libraries on beads?
Yes, actually it can be. The only thing is, so the solid phase synthesis, it actually Bruce Merrifield introduced like 1960s, and he won the Nobel Prize also in 85. It has been very popular method in synthetic organic chemistry, but not all the compound being transformed into the solid phase. So this peptide, peptoid, aptamers like chains can be synthesized very easily, residue by residue adding. For small organic molecules also it can be used, but not in a very like large scale as we do for other compounds. So there's kind of a limitation for small organic molecules to be synthesized on bead. But to give you some of my work also, which I didn't present, we actually take like daughter-like molecules and actually modify those side chains, like branching out, which is like developing some library of organic molecules. So it can be done, but not in a very large scale. So that's kind of a limitation there. The
Right.
peptoids also, as you said, I mean, no single molecular class can do everything. So the peptide solid phase, this screening approach can be used not only in peptoids, but definitely peptides, aptamers, like DNA, like type libraries.
Great. Thank you, Prof. Udugamasooriya. And then we have a question from Prof. Tilakaratne. Great presentation, Gomika. Many drugs candidates fail because of their effects on HERG channels. Does your assay screen for effects on HERG channels?
Yeah, wonderful question. So yes, actually, absolutely. So I mean, there are so many collaborative requests coming to me. So the bottom line is, if you have a biomarker on cell surface, we can actually go behind it. So this is definitely a possibility. Only thing we need is two cell groups. One has the target, the receptor, the other don't. So it can be achieved in two ways. One is we can take the cell line, divide it into two groups and overexpress the target. Or we can take a cell line that has the target, and we can knock down the protein and then produce the other cell group without the target. Yeah.
Thank you. And then we had another question on the chat asking about your thoughts on using AI and how AI can be used to improve the outcome of similar researchers.
Yeah, so everybody's talking about AI, and of course, I mean, again, so I'm always thinking like unconventional ways. So that's how I apply this also in a very unbiased fashion. But AI actually it's data driven, but it trains. So probably, I don't know, we can think about designing libraries in a more biased way, meaning like um, look at the possibilities of targeting. We can select side chains based on some information and looking at everything available, what kind of chemical properties may be needed for the libraries. Then we can select the amines that are kind of biased towards again, even it can be used to target a certain disease types, and based on uh, the available data and applying AI. So there are so many possibilities. Yeah.
Thank you. So we have another question from Angelo about labeling the cells. Are you internalizing the colors, probably fluorophores or any other form for colors, or are they surface modifications?
Excellent question. So no, they are actually internalized. So first of all, I was very careful not to touch the surface because that's what we are going to target. And then secondly, I mean, I mentioned before I bring this two color method, the the screenings were done just one step without these dyes. There was a technical barrier also. If you use organic dyes, these beads also have a long uh, emission spectra, so the bead also get that color. So if you use FITSI, the bead also become green color. So you cannot use the way we address that issue was we use quantum dots. So the quantum dots actually excite on the low level and express far away. So the images you saw are all three colors in one shot. It's not overlaid. So in a DAPI a long pass filter. So again, yeah,
Internalization, yeah.
it's internal.
Yeah, thank you. Thank you very much. If you have any other questions, please unmute your microphone and ask the question. Well, I actually have two questions, Professor Udugamasooriya. The first one is actually on the COVID project. The compound seemed to be very effective and at the same time selective too. I was wondering if you had the chance to do any actual functional assays to show the toxicity on kidney and liver side of the physiology?
That's what we exactly doing now. So I mean, in animals, we did these studies in hamsters. So there are no, I mean, we look at the standard body weight and the other symptoms. But yeah, we are right now actually we are working on this full toxicology studies.
Great, thank you. And the other question I had was on the cancer stem cell side, which is my interest of our studies as well. So the cancer stem cells were selected, if I understood it correctly, based on the expression of ALDH,
Yeah.
aldehyde dehydrogenases.
Right.
So I was wondering the method, what if the cancer cells, some cells, they do not have the overexpression of ALDH, but other sort of cancer stem cell biomarkers. Do you think that's going to be applicable for those kind of cancer stem cells as well, or?
Absolutely. Yes. So this is the biggest problem, right? The heterogeneity. So
Yeah.
the reason we use LDH because that's the assay kit is commercially available. This is a standard method. So we try to be as close as to the standard method. So once again, to answer your question precisely, we cannot claim it's the perfect way. There's no perfect way to do that because the biomarkers are very heterogeneous and there are many subpopulations there. Again, we can apply to those different ones separately. That's another good application.
Yeah. Okay, so one last question from the chat. Are there methods being explored to prevent the cell membrane lipid flipping that allows the cancer cells to go undetected?
Prevent the cell membrane lipid flipping that allows the cancer cells to undergo actually it undergo undetected because of the flipping. When the that lipid come to the surface, it's a stop sign for the natural killer cells to kill it. So it shows the stop sign when the flipping happens. So I think the question was meant to, can we stop the flipping, right? So then it can be detected. That's what we are doing actually in our compound, masking that PS coming. Of course, there are mechanisms, enzymes, everything has been identified for this flipping. I think there are a lot of work going on to block that as well.
Great. Looks like we don't have any more questions. That was a wonderful, wonderful talk. Thank you very much for being with us today. So we always would like to hear from you, and please let us know if you have any suggestions and comments by contacting CAPSA Speaker Series Subcommittee using the email address shown here. Also, let us know if you have any suggestions for any specific topics or speakers that you would like to see in future. So now I would like to invite Professor Hasini Pereira from the Department of Chemistry, University of Colombo, to deliver the vote of thanks.
Okay, so let me take this opportunity to thank Professor Gomika Udugamasooriya on behalf of the Department of Chemistry, University of Colombo, on delivering a very interesting talk on novel technologies to expedite the drug lead discovery process. We are all indeed fortunate to hear about all this ongoing research carried out under Professor Udugamasooriya's supervision. I especially found the advanced stages of animal studies very exciting. And on behalf of the University of Colombo as well as the Department of Chemistry, we would like to wish Professor Udugamasooriya all the very best in your journey towards the drug lead discovery. And thank you again for spending your valuable time to educate us. And I'd like to take this opportunity to thank the CAPSA distinguished speaker, the series as well, for allowing us to be a part of this wonderful talk. Thank you very much.
Thank you, Professor Pereira.
Thank you so much, Hasini, Vasundhara, and CAPSA and the Chemical Society of University of Colombo. All the best to everyone.
Also, we partnership with the Chemical Society of University of Colombo when bringing this talk to you. So we especially thank them as well. And Professor Udugamasooriya, one more time, thank you very much for joining us and then giving this wonderful talk. We have now come to the end of today's seminar, and on behalf of CAPSA, I would like to thank all of you for joining with us today, and stay tuned for our future talks through the CAPSA seminar series. Now, we will be concluding today's session